Hello everyone, welcome to the final presentation of our iGEM Indonesian League project titled Developing a Nepetalactone by Synthesizing Carbon Assimilating Yeast. My name is Michael Gita Nobel and I'm the team captain of Menadoensis. Menadoensis is originally a scientific community made up of past winners of the Indonesian Biology Olympiad and the International Biology Olympiad. We are very grateful to have Alfred and Jose as our advisors and also our external advisor, Dr. Aditya. I want to also thank every member of the Menadoensis team for their contribution to the team and the project. Our team is very diverse, composed of people from 8 universities in 4 different countries. These are the special awards we aim to reach. The details of each prize will be touched on in each segment. First, let's talk about our project background. We have seen many news outlets reporting the current air pollution conditions in major Indonesian cities. For example, BBC News reported that Indonesia is one of the 6 countries that contributes the most towards global air pollution. The deteriorating air quality in Indonesia is reflected by major Indonesian cities such as Jakarta, topping the world ranking for the worst air pollution. Other regions in Indonesia such as Palembang also experience a similar problem. Vehicles using fossil fuels contribute a lot towards these air pollutions. However, the ongoing wildfires in Indonesian forests also contribute to air pollution. The combustion of organic matter such as fossil fuels and trees releases carbon dioxide. The carbon dioxide is then released into the atmosphere. Unfortunately, carbon dioxide is one of the greenhouse gases. These gases create the so-called greenhouse effect, in which gases in the atmosphere trap the radiating solar heat from being reflected away into the space. Carbon dioxide itself is the single largest contributor to the greenhouse gas reservoir, almost 80% of all gases. The long-term entrapment of the solar heat causes a rise in global average temperature, with a rise of approximately 1 degree Fahrenheit each decade. The temperature rise also causes the glaciers in the Earth's pole to melt, which subsequently causes the sea level to rise. We have probably heard that Jakarta is sinking due to the excessive use of underground water supplies, but global warming will help the city to sink even faster. Global warming also affects the distribution of animals and plants, including disease-bearing organisms such as mosquitoes. Because they live in hotter and arid regions, the increase in surface temperature widens the distribution of mosquitoes. This graphic illustrates the increasing possibility of disease transmitted by Aedes aegypti, a species of mosquito, throughout the globe. These diseases cause significant financial and healthcare problems. For example, dengue fever inflicts a staggering $8 billion loss to the world economy.
Pichia pastoris, a methylotropic yeast, has emerged as a robust platform for heterologous protein production and metabolic engineering. Its ability to efficiently utilize methanol as a sole carbon source has garnered significant attention, particularly in the realm of sustainable biomanufacturing. To further enhance its metabolic capabilities, we embarked on engineering Pichia pastoris to incorporate the Calvin-Benson balsam, or CBB cycle, enabling carbon dioxide assimilation. We also introduced nepetalactone synthesis pathway, which makes it possible to produce nepetalactone. Nepetalactone will act as an insect repellent, and hopefully the mosquito problems will be solved.
Because we seek to tackle 2 problems at once, and because of the significance of our project, we believe we deserve to be awarded the Best Local Problems Award. To introduce our solutions to a wider general audience, we created a trailer video that can be accessed through this QR code. Because of its quality, we believe we deserve to be awarded the Best Trailer Video prize. We put utmost care into ensuring that our project complies with biosafety and biosecurity regulations. Therefore, we consulted Dr. Richard Burke, a member of Monash University's Biosafety Committee, in preparing our safety form. Most of the genes we use in this project come from P. pastoris as our chassis organism and Nepeta cataria, both of which are relatively safe organisms. Therefore, a biosafety level 1 facility is sufficient to execute this project. To prevent contamination of the native P. pastoris gene pool, we incorporated a kill switch to kill the cells that escape a microenvironment that will be provided by a bioreactor housing these cells. Nevertheless, even if a spilled cells survive, they have an evolutionary disadvantage compared to wild-type cells due to the genetic burden of the incorporated genes. Before we can realize our project, we will need to seek permission for dealings involving international release from the Office of the Gene Technology Regulator. For our work in maintaining biosafety in our project, we believe we deserve to be awarded the Best Safety Practices prize. Now, we discuss our workflow. The full documentation can be accessed through this QR code.
Naturally, PCA use XUMP cycle to produce biomass from methanol. With the XUMP cycle having several key Calvin-Benson balsam cycle enzymes, we thought of introducing a CBB cycle to enable PCA to assimilate carbon dioxide. To introduce this cycle into Pichia pastoris, we carefully selected enzymes from other organisms known for their optimal performance in this yeast, such as Rubisco from Thiobacillus nitrificans, along with its chaperone proteins, GROEL and GROES. Concurrent with carbon dioxide assimilation, we sought to establish a nepetalactone production pathway within Pichia pastoris. Nepetalactone, a naturally occurring compound found in catnip, has garnered interest for its potential medicinal and insecticidal properties. By using the glycolytic and mevalonate pathway, we enabled the conversion of glyceraldehyde 3-phosphate or GAP into isopentenyl pyrophosphate or IPP, which then will be converted into nepetalactone. To maximize nepetalactone production, we employed targeted enzyme optimization strategies. These strategies included mutagenesis of the ERG20 gene. By disrupting this pathway, we effectively diverted metabolic flux towards nepetalactone production. Additionally, we introduced multiple copies of SMGR and IDI1 genes from other organisms, further enhancing the efficiency of nepetalactone synthesis. The engineered genetic construct was integrated into Pichia pastoris genome using CRISPR-mediated homologous recombination. We distributed a total of 22 transcription units into 4 BB plasmids that will be integrated. This technique involved replacing the AOX1 gene, DAS1, DAS2, and ERG20 segments of Pichia DNA with corresponding segments from the 4 plasmids harboring the enzyme coding sequence. To ensure controlled survival of the recombinant organism, a tetracycline inducible killing mechanism was incorporated. In absence of tetracycline, the transcriptional activator ceases to function, preventing the production of the antitoxin. This leads to cell growth, inhibition, and subsequent cell death. This safety measure effectively limits the unintentional persistence of the engineered organism beyond the desired experimental condition. Through these metabolic engineering efforts, we successfully transformed Pichia pastoris into a carbon dioxide fixing nepetalactone producing organism. This achievement holds promise for sustainable nepetalactone production and demonstrates the remarkable potential of Pichia pastoris as a versatile platform for biomanufacturing.
In literature review, we found nepetalactone is able to bind with transient receptor potential and kirin-1, especially with mosquitoes TRPA1 and not to human. Therefore, we would like to explore more about interaction between Aedes aegypti TRPA1 or TRPA1 and nepetalactone with other compounds as well. Based on articles data, we discovered that isoform G has property to bind well with nepetalactone, and by looking to the structure, we predicted that there is a region responsible for this interaction. Exploration was done in PDB database, and we found that there were 6 variants of ATRPA1 with respective properties. After found the resource in PDB, we tried to match between the online database and other journals, and we conclude that nomenclature of variants used in previous article was same with PDB nomenclatures. Then we downloaded their sequences and did multiple sequence alignment into cell omega. The result was visualized in Mega 11, and we studied that isoform G has residues differ from other variants of region we predicted before. Interestingly, isoform B also has the sequence of interest. From previous study, isoform F was unable to respond to any kind of stimuli, so we determined to use 3 isoforms in our study, B, G, and F, where isoform F acts as negative control. From mole probability, we received respective property results, concluding that their structure were good enough for molecular docking analysis. We visualized 2 of our receptors of interest in Chimera and discovered that sequence of interest determined previously is an alpha helix, and it's located near proteins and kirin domain. Then we prepared our ligands based on our literature review. We chose 5 ligands in our study, respectively. By their molecular property, our ligands have low molecular weight and high lipophilicity. Thus, they could easily pass cellular membrane to interact with transmembrane region of interest in the receptor. Based on our study and preparation, we predicted that alleles of isocyanate, AETC, and nepetalactone should have good interaction with ATRPA1, and other ligands should have less interaction. We also divided our experiment into two groups, where the first group using ligands without energy minimization and the second group using energy minimized ligands. We predicted that there should be difference in result between those two groups. Molecular docking was done in Pyrex and recorded respective binding affinity values. The results are listed in respective tables. The lower the binding affinity value, the better predicted interaction between ligands and receptors. Therefore, it was clear that isoform F as negative control had lower binding affinity with respective ligands compared with other isoforms. Docking visualization then was done in BioVia Discovery Studio. We reject our first hypothesis by the result that ETC had the lowest binding affinity between all interactions. This might be because of previous articles stated IETC interact with covalent bond, but our tools were just able to visualize non-covalent interaction. We accepted that nepetalactone had good binding affinity with the receptors, especially isoforms G and B on the region of interest, proving our second hypothesis. Otherwise, even the rest of ligands showed lower binding affinity than nepetalactone to ATRP1, they still had higher binding affinity than ATC. Thus, we reject the third hypothesis, and we found unique citronellal interaction. And this discovery open future chance to research more about ATRPA1 interaction. At the end, we agreed that minimized and non-minimized ligands show difference in their binding affinity values, accepting the fourth hypothesis.
We did several optimizations for this project. First, we chose several methanol-inducible promoter to control our CBB cycle genes. We also chose promoter gap, a constitutive promoter for nepetalactone synthesis pathway gene to ensure continuous production of nepetalactone. Next, we mutagenize ERG20 genes in the 98th and 128th amino acids to bias it towards nepetalactone production. Then we did codon optimization for all the introduced genes and removal of BSA1, BPI1, and SEP1 restriction site. These steps are required to ensure efficient translation and compatibility with Golden Gate assembly. We added peroxisomal targeting sequence for CBB cycle genes to localize the CBB cycle in peroxisome. We also implemented a prion-based scaffolding system for nepetalactone synthesis pathway. Finally, we construct a bubble column reactor to ensure high level of carbon dioxide in the medium. We also added tetracycline to ensure cell viability and methanol as an additional carbon source for Pichia pastoris. In this project, we applied several synthetic biology principles. First, we ensure the compatibility of the parts used in our project with iGEM RFC 1000 standards by eliminating all BSA1 and SEP1 resisting sites. We also adhere to the modularity principle by constructing BB2 plasmids for transcription factors and BB3 plasmids for multi-gen construct used in this project. We adhere to the principles of predictability and control by constructing genetic circuits for all genes used in the project, which will simplify further metabolic and mathematical modeling. Iterative design principle is applied in TRPA1 anti-mosquito compounds molecular docking by formulating experimental procedure, doing molecular docking, and analyzing the result. We repeated these steps multiple times with improved procedure based on the obtained results. As already mentioned before, we did several optimization, including promoter choice, codon optimization, standardization, molecular docking analysis, and many more. To minimize the ethical issue with our project, we did several steps we mentioned earlier to ensure the biosafety of our project. For interdisciplinary collaboration principle, we intend to collaborate with the researcher with strong background in mathematics and programming for further metabolic and mathematical modeling. Finally, we ensure data availability of this project by uploading all experimental data into BenchLink, which can be accessed by following link.
Wow, elaborate work, wasn't it? That's why we believe that we deserve the best project report award. Besides all those works, we performed an extensive attempt to demonstrate human practices on our project. We launched a segment called Expertise on our YouTube channel and Instagram page. We discussed our project with 6 international experts with expertises related to our project. There are 6 episodes of expertise. We interviewed Dr. Carlia, Dr. Almando, Professor Victoria, Dr. Adityo, Professor Reskumar, and Sura. Next, we organized an online symposium titled Synbiotechfest to discuss issues around synthetic biology and the Biology Olympiad. We aim to introduce the realm of synthetic biology to high school biology Olympiad talents to pique their interest in the application of biology. Our symposium successfully raised 500,000 rupiahs for the World Mosquito Program, a project to eradicate mosquito-borne diseases by using Wolbachia-infected mosquitoes. We also launched a special series of infographics on our Instagram page, which disseminates our project and explains multiple topics in synthetic biology. We also partner with Tam Patisena Institute, an Olympiad preparation institution, to teach the students about molecular genetics and the basics of synthetic biology alongside our project. For all our extensive attempts, we believe we should be awarded the Best Human Practices Prize. To conclude, we re-engineered the Pichia pastoris into a carbon-assimilating organism by performing genetic modifications. We optimized the gene sequence and incorporated a kill switch to meet biosafety principles. We also combined the biosynthesis pathway of naphthaloctone, which has been found through molecular docking to elicit aversion response in insects, with the glycolysis and the methylonate pathway. You can find our social media here, including our Instagram page and YouTube channel. If you have any questions, please direct them to our email. Thank you very much for your attention and have a great day. See you at Synbiotechfest 2023.